Okay, so what I'm going to talk to you about today is work that we've been doing in the lab. It's not just me. I work with a lot of different students and postdocs and collaborators. But this is the work that I like to drive, and it's understanding the evolution of large brains. And so what I want to go through is static. How do you actually, if I go like that, that's better. Okay, I don't get static shock there. What I want to talk about is how we define scientifically what a large brain is. The fruit fly has a small brain, the human has a large brain, but does a monkey have a large brain? In comparison to the fruit fly, it does. So how can we say what a large brain is scientifically, using a real basis for that? And defining which species actually have large brains once we have that definition. To show that there are several evolutions of large brains in mammals and that these all followed independent evolutionary trajectories. And to show that, for the most part, allometry of body mass is the major factor in evolving a large brain. Nothing to do with intelligence. Okay, so I'm going to try to avoid as much as possible a whole idea of intelligence today. The next thing I want to show is that there is a specific metabolic adaptation that is the key for evolving a large brain. This is new and unpublished work that we have been doing, and some of the stuff we only found a week or so ago. And then to show that this supposed need for greater intelligence is not a factor in the evolution of large brain size. The human brain, the whale brain, the elephant brain, the camels and the seals, none of these brains evolved their large size for intelligence. Okay, so that's what I really want to get across through this talk. Now, this picture is one of my favorite pictures to show because it shows a really important concept in evolution that many people misunderstand, especially in the neurosciences. So this, especially for those from anatomy, will know that these are the bones of a human arm. Those who have done a bit of comparative anatomy will know that these are the bones from the flipper of a very large whale. They're homologous. There's a humerus here, ulnar and radius, ulnar and radius. I've even forgotten the bones on the wrist. It's been so long since I've had to teach gross anatomy. The carpals, and the other bones. We know they're homologous. We also know that the human hand is very manually dexterous because we can put two fingers together. Some of us better than others. I'm not so good with the little finger. That means that this hand has evolved and is specialized for manual dexterity. Because this arm is larger, does this mean that this hand is more manually dexterous than this hand? And the simple answer is no, because they're built differently. They've come along different evolutionary trajectories, and they do and accomplish different things. So just because a structure is homologous, it doesn't mean it does the same function. Homologous structures are not always analogous in terms of their function. And this applies very strongly to the brain. So with that in mind, I want to continue now into some of the more mathematical aspects of brain evolution. This is a plot of body mass versus brain mass across a whole bunch of mammals, a bunch of primates, and some of our fossil hominid ancestors. Now, the first thing I want to point out is that for mammals, there's a beautiful regression between body mass and brain mass. And you can see that the probability that the size of the brain is growing independently of the size of the body is 4 times 10 to the minus 294. I don't even know what that is in words like billions or trillions, but there's 1 in bazillions chance that these things are not correlated. And in fact, if you look at the R squared here, this tells us that 94% of the variance in brain mass across mammals can be explained by variance in body mass. Okay? So 94% of the changes in the size of the brain are a direct result of changes in size of the body. If we look at primates, a similar regression occurs, a little higher. Primates have brains that are about twice the size of a mammal for their, twice the size of what you would expect from a mammal of their body mass. And we see the correlation again is very, very high, and the explanatory value of this regression is also very high. So the really important point here is that brains evolve larger size mostly because of body mass. That's really the important point. But then we reach a point where things get a little unusual, and this is up here. And this is something that I've been able to contribute to. We have data points from rhinoceroses, hippos, camels, giraffes, and other things up here. And we see while this regression stays very nice for a long way, it starts to taper off. And there's like a ceiling here, a ceiling at around 700 grams. It means that generally for mammals, brains can't get bigger than 700 grams. And this is kind of like a glass ceiling on evolution of brain size, which is really quite unusual. And we see even here in the hominid lineage, we have a bunch of hominids with small brains, and then a few that have large brains. These are the elephant data points up here. But I'll go into more detail here. So the 2 points I wanted to get across in this slide, and they're the important points at the start, is that body mass predicts brain mass 95% of the time, and that there's a limit of around 700 grams. This here is a hippo that weighed about 2,500 kilograms that I got at the Copenhagen Zoo, and it had a brain of 530 grams. So even though it's an enormous animal, it has a brain that just can't get any bigger than 700 grams. So there are animals that do get brains larger than 700 grams. And it's limited to mammals. The largest bird brain is 35 grams in the ostrich. So when we're talking about large brains, we're talking about mammals. The majority of the genus Homo, but not every representative of the genus Homo, extant elephants, extant cetaceans, there's 3 pinniped species and 1 camel species. And all these groups have, all these brains are very phylogenetically distinct. They're structurally distinct brains. So there's 7 different trajectories that led to the evolution of a large brain. And I want to look at these in a row. We'll go very quickly through the pinniped and camel species because I don't have a lot of data on those, but I have enough to explain what's going on. Then spend more time on humans, elephants, and cetaceans, and then try to bring it all together at the end. So this is a picture of a walrus brain. The walrus brain weighs about 1,160 grams. It's a good sized brain. If you were Philip Tobias, you would say this is large enough for this animal to have language. That's what he said about hominids. But when we look at seals, there's actually 3 species of pinnipeds that have large brains. We have the elephant seal, the walrus, and the leopard seal here. Now here again we have body mass plotted against brain mass. We see a nice regression, very nicely statistically supported and so on, amongst the seals. This is just data from seals. And we see our 3 animals here. We have prediction and confidence intervals. And these 3 seals, despite having brains over 700 grams, fall well within the statistically defined confidence intervals for this regression. So we can say that for the most part, the reason that these animals have large brains is scaling or a long tree with body mass. If we look at the Bactrian camel, this is the Bactrian camel. This is a Bactrian camel brain that actually again came to me from the Copenhagen Zoo. My friends there helped me out a lot with these specimens, and it's a nice big brain, 730 grams. It's just creeping up over that 700 gram limit. If we look at a regression for artiodactyls here, we see the brain falls very close to the regression of body mass or brain mass here. So again, we can say that even though this animal is over the 700 grams, the brain has a brain over the 700 grams. The reason it has the brain this size is based on an increase in body mass. Now what about human brain evolution? This is a different one. This is my son, by the way. Am I the descendant of a monkey? And of course, we all know that direct lineage isn't working out. But how did humans get larger brains than other primates? And if we look at the regression again, body mass against brain mass, we see non-human primates have a very nice regression, very strong statistically supported and very high explanatory value. Then if we look at the hominids, and these are all the animals that are found in the fossils that are thought to be in our direct ancestry. Not direct, well, within our ancestral group of humans. So the Australopithecines, anything that's considered Homo and so on. And again, we get a beautiful regression there. We have a regression that is strongly statistically supported and high in explanatory value. But there's something quite different between the primate regression and the hominid regression. Primate regression has a slope of 0.76. That means it's negative allometry. So that for every doubling in body mass, the brain increases by about 1.3 times in size. When we look at the hominid regressions, we see that the slope of the line is 1.63. That means that for every doubling in body mass in the hominid lineage, the brain gets bigger by about 2.5 to 3 times. So this is, and it's called positive allometry because it's over 1. So the brain scales up much more rapidly than what we'd expect based on the other mammals. So when we look at human brain evolution over time, we can say, all right, we have the mammals as our baseline. The brain gets larger in primates. Primates have large brains to their body mass compared to other mammals. The other thing that primates have that's really interesting is not only is their brain twice the size to their body mass compared to other mammals, they have twice the density of neurons, or twice the number of neurons you would expect to their brain size compared to other mammals. And this is an important point because this will lead on to understanding why humans have the cognitive abilities we have. So at the origin of primates, there was something that shifted in a quantitative way the allometry between brain size and body size to a seeming advantage in primates. And they had extra neurons. Then when we look at the start of the hominids, there's another factor that changed the relationship between brain and body scaling. And I've put in question marks bipedalism, because this seems to be the largest, or the most significant change in the evolution of the hominids. So when we put all this together, we can summarize the evolution of the mass of the human brain. For the most part, the mass of the human brain is associated with scaling of body mass, going from a negative allometry to a positive allometry. There's 2 possible adaptive events at the origin of primates and at the origin of hominids. These changes may be positively selected changes. They may be adaptations, or they may just be neutral changes, things that happen that then show a change later on. So bipedalism is a great example of this. It may have led to an altered scaling of the brain-body relationship, but at first that might have been neutral. And I want to show in the next slide why I think this is neutral. But then later on, it may have caused major changes in the evolutionary history of the hominids. So if we look at human brain and body mass, or hominid brain and body mass over time, I call it the Great Leap Forward. Here we have millions of years before present. We have body mass. We have the Australopithecines and Paranthropus, Homo habilis, and so on, several million years ago. Then there's a big jump in body mass to Homo ergaster. If we look at the cranial capacity of these animals, so the size of the brain in a rough sense, again, we have the same thing, a lot of stasis followed by a big jump, then eventually the brain increases and the body mass increases. And see, the brain mass increases much more steeply than the body mass because this is positive allometry. So the important point here is that there was stasis. After our ancestors were bipedal, For a long time, there was stasis, many millions of years, stasis in body mass and stasis in brain mass before something happened, and then bodies started, bodies started to get bigger and brains started to get bigger in a very rapid way. Okay? So does this alone explain the cognitive capacities of humans? And I don't want to get into an argument about the intelligence of humans. We know there's a lot of unintelligent humans out there, as we like to put it. But in general, humans have a cognitive capacity that's far beyond even our closest relatives. So can we say that size alone accounts for the cognitive complexity of modern humans? To do that, you have to actually start looking at the building blocks of the brain. And these are the neurons. So these are pictures from a series of experiments I did not long after I got to South Africa, where we got baboons, vervet monkeys, and other species. And these are filled cells, layer 3 pyramidal cells. And this is a 3-dimensional reconstruction of one of the cells that we filled. And we can see a whole range of features. We can see the cell here where we put a pipette in and pump the dye in. We can see the size of the dendritic field, and on the dendrites you can see the spines and so on. These are the important parts in terms of communicating amongst neurons and building particular behaviors. So these are taken from the cortex. We actually looked at about 6 or 7 different primate species, including data that we got from humans in Spain. In Spain, they have a rule that says, say you get killed in a car accident, your brain can be taken to scientific research straight away. And so there's really, I don't know if it's an advanced way of thinking, but it's a different way of thinking. So we looked at neuronal complexity. We measured the size of the basal dendritic field area of the pyramidal cells. That gives us an indication of the amount of information and the differentiation of, or the amount of different information that an individual neuron can pick up. The larger the basal dendritic field area, the more information it can pick up. We looked at the extended dendritic branching. This is important because in dendrites, there's a dendritic current that flows from the tip of the dendrite back towards the cell. And this dendritic current is what causes things within the cell to change and action potentials to form. The more branches you have, the more complex and more local processing can happen before information reaches the cell. And the last thing we have is the numbers of spines. So the numbers of spines are where the synapses come onto. So you can imagine cells with lots of branches and lots of spines and a large basal dendritic field area can actually absorb a lot of information and use that information in a way to generate action potentials in a very particular way. Sometimes more complex, sometimes less complex. So we looked at animals from galagos to humans and looking at this dendritic complexity in a few different cortical areas that we knew were homologous across species. And what we found was that the complexity of the neurons scaled elementrically with brain mass, but not in all areas. For example, in primary visual cortex, there was no scale. The complexity of the neurons in a galago primary visual cortex are basically as complex as what you find in a baboon or a human. But then when you move into higher order visual areas, V2, V4, area T, which is a facial recognition visual area, we see there's allometry. So the neurons are becoming more complex as the brain gets larger. And then when we look at, say, area 10, which is part of the prefrontal cortex, you see massive changes in the complexity of the neurons between humans and galagos down here. So there's a few things that are going on in the primate brain. First of all, we have twice the number of neurons of other mammals and primates. And secondly, as primate brains get larger, the neurons become more complex. So obviously humans are kind of the endpoint of this. So human behavior is more complex due to an increased complexity at the level of the neuron. We have a greater number of more complex units than are found in any other mammal. That's a very big difference. The neurons are more complex because of increased brain size, and brain size increases in parallel with increased body size, and at the origin of hominids, a change from a negative allometry to a positive allometry. Those three things put together help us understand human brain evolution and what it means for human brain evolution. So the majority of human brain evolution and our resultant behavioral complexity can be explained by allometric scaling laws of form and phylogenetic constraints. We're complex because we descended from primates. That's the main reason that we're complex. And primates have these allometric scaling laws and phylogenetic constraints written into their brains, into their genes, I suppose you could say. We don't need to have adaptationist narratives. We don't need to trade off gut size. We don't need to have fat babies and all these different things. The only time we need to actually invoke any adaptationist thinking is at the two specific events, the origin of primates and then at the origin of hominids. Other than that, there's no need to resort to complex and sometimes very convoluted ideas regarding human brain evolution. My answer to this question is no. I don't think intelligence was selected for in human brain evolution. I think it just kind of happened. All right? So that's humans. Let's go on to elephants now. So I'm going to change track a few times during the talk. So we look at elephant brain evolution. This is a nice picture I managed to take in the Kruger Park before we had to drive away very fast. So we look at the trajectory of elephant brain evolution. The origin was in the Eocene for the proboscids. It's a small pig-sized mammal. There's increases in body and brain mass over time. The trunk length increased, tusk size increased. They had 4 tusks originally, then they went back to 2. But the relative brain size of modern elephants is 1.2. It's about what you would expect for a mammal of its body size. Okay? So we're getting back to allometry again. Sorry, I'm going to talk a lot about allometry. If we look at elephant body mass over time, we see something very similar here to what we saw in the hominid lineage. A period of stasis in body mass, followed by a massive jump in body mass and then diversification. Some of the animals were absolutely huge. It would be interesting to get ideas of the size of these brains. I think one of my former postdocs, Julian Benoit, who's in the Institute for Human Evolution, would have a lot more answers about elephant brain evolution than what I do. But the point is that there was a lot of stasis. Now, I said to you that allometry is the thing that really predicts brain size across mammals. So if we look at elephants, we follow the allometry up here, and you can see if we had 95% confidence intervals, then they'd fall right in it. And actually, in the next slide, I do have that. So we see this for mammals, we see the confidence intervals here. This is extant elephants, plus some of the more recent extinct species. And here is an archaic elephant plotted against archaic ungulates, which are perissodactyls and artiodactyls. And we see here at the time that this Neotherium leonsoni was existing, it scaled very similarly to other mammals at the same time. So it appears that over their evolutionary history, the relationship of the brain to body mass in elephants has always followed the predominant scaling of the relationship of brain to body mass in mammals in general. All right? There wasn't anything particular selecting for a large brain size in elephants, although after a period of about 40 million years, when there was a lot of stasis, and presumably brain size stasis in this as well, was followed by a massive jump in body size. And I assume here that there was a massive jump in brain size. And that follows the mammalian allometry the mammalian allometry of brain to body mass. And that accounts for the larger size of the brains in elephants. Elephants have about 5 kilogram brains, so they're quite sizable chunks of neural tissue. So again, there's nothing there that says these guys needed a large brain for intelligence. Okay? We have done work on the neuronal complexity of elephants. There are some neurons in the elephant brain, we call them matriarch neurons, that are as large as the most complex neurons in human brains, but those are reasonably few and far between. Compared to the human cerebral cortex, the elephant has about 5 billion neurons in its cerebral cortex, the human about 16 billion neurons, and the elephant cerebral cortex weighs about 2.5 times that of the human cerebral cortex. So they have a much lower number of neurons in their cerebral cortex, despite it being larger. But they do have some neurons that have a reasonable amount of complexity, which I think underlies a lot of the behaviors we see in elephants today is based in this complexity of their brain and the size of their brain, but I don't think that was selected for in elephant brain evolution. There's no evidence to say these things were selected for. It really looks like they follow all the allometric rules of mammals in general. All right, let's get on to the group that I'm infamous for, cetaceans. And I picked this picture particularly because of these little guys here, which are photoshopped in, by the way. Lots of people think whales and dolphins are second to humans in intelligence. So what happened in cetacean brain evolution? First, if we look at the allometry of extant cetacean brains, we see our mammalian regression here, our primates, our hominids, and here's the cetaceans. And it's very, very different. It's a very shallow negative allometry. Some of these animals have relatively large brains. Some of them have relatively small brains. So using the idea of relative brain size doesn't explain anything really about cetaceans. You've got some that have got relatively small brains, some that are relatively large. It doesn't work. You can't equate that with intelligence. So when did cetacean brains become large? This is a series of regressions from archaic ungulates, which are artiodactyls and perissodactyls grouped together, coming up to the recent artiodactyls. These are the Archaeocidae. So the Archaeocidae are the ancestors of modern cetaceans. They are fully aquatic animals, and they lived for about 20 million years, from about 53 million years ago to about 35 million years ago in shallow seas around the equator. Then their direct descendants, the Oligocene Odontocetes, Miocene Odontocetes, and the extant cetaceans, all have very similar scaling of brain mass to body mass. And we can see that the relative size of these brains increased. These are the ungulates over time with slowly increasing relative brain size. Here's the Archaeocidae, which match these animals that were around at the same time in terms of mammalian scaling. Suddenly there's a huge jump. We have much larger brains relative to body mass in cetaceans. And not only was there a huge jump in brain size, this scaling has stayed the same ever since. So it hasn't changed. There was a change, and then it stayed the same ever since. The other thing that happened was the relative proportions of the brain changed. So this is a picture of an Archaeocidae endocast. It was actually given to me by Professor Tobias, who inherited it from Raymond Dart. So all the way back to Brisbane, where I came from. We can see in a drawing of this, you can see here's the cerebral cortex, the cerebellum. It's flipped around mirror image, trigeminal nerve, olfactory tract going down to the spinal cord. But I wanted you to notice, here's the 1 centimeter scale bar. This is the cerebral cortex. It's about 5 by 5 centimeters. It's pretty small. This animal was 14 meters long. It was a huge animal. And it lived in the Tethys Sea, which was the equatorial sea before India crashed into Asia, Africa closed in on the Mediterranean, and North and South America came closer together. The Archaeocidae were living in this sea with these small cerebral cortices, nice big cerebellums with small cortices, for about 20 million years. Then they died out, and the modern cetacean flora appeared. And here's another fossil endocast from the Miocene, and we can see the shape of the cerebral hemisphere here, which is very similar to the cerebral hemisphere of a modern dolphin. We're looking at that's from above. We look from the side again. We can see this very typical sort of boxing glove shape of the cetacean brain from the side. So with cetacean brain evolution, a few things happened. There was a dramatic increase in relative brain size and proportions of the brain. This was then followed by stasis. There was a change in the scaling relationship between the body and the brain. The negative velometry became even shallower, so that every doubling in body size in the cetacean only leads to about a 1.1 to 1.15 increase in brain size. There was a loss of the previously warm, nutrient-rich Tethys Sea, and there was a cooling of global oceanic temperatures. So if we look at a million years before present, so here's the Archaeoceti. They branched to give the Mystaceti and the Adoniceti, which are the modern cetacean fauna, and here we see a massive drop in the deep ocean temperatures. And this coincided with India crashing into Asia, Africa coming much closer to Europe, and North and South America coming together, and the Tethys Sea being wiped out. The whole globe cooled down dramatically. Now, this drop was about 6 degrees in deep ocean temperature. A lot of people say, well, 6 degrees, it's not that much. But remember, the temperatures they're living in are around 15, 16 degrees normally anyway. So I defy any of you to stand naked in 10-degree water for more than 10 minutes. I don't think anyone could do that, right? It wouldn't be so good. So to me, this is a very, very strong selection pressure. And this is where I developed the hypothesis that when they lost the Tethys Sea, which was warm and nutrient-rich and shallow, the oceans became cooler, that this change in temperature was the driving force behind evolution of large brains in cetaceans. And it also changed the way that their brains scaled. So the cetacean brain enlargement as a response to combat heat loss in water. That's the hypothesis. So now when I came up with that idea, then I have to prove it, of course. So I'm not going to go through all the details, and I'll give you some of them. This is a relationship of relative brain size to the range of water temperatures cetaceans live in. And there's a very nice allometry here again. The greater your encephalization or your relative brain size, the greater the range of water temperatures you can live in. It has nothing to do with absolute size or absolute brain size, absolute size of the animal or absolute brain size. It's all to do with relative brain size. So this indicates that really this relationship between brain and body is incredibly important. It's something that a lot of people have ignored over a lot of the history because we want us to think about intelligence. All right, so I'm going to come back to that in a couple of minutes. So what I hope I've shown you over the past few minutes is that there really are 7 or more distinct pathways to evolving a large brain. There's many differences in the structure of the 7 large brains. We can't say that a camel brain and a walrus brain are the same thing. A camel brain evolved from an artiodactyl, a walrus from a carnivore. Their brains are structured differently. So there's minimal phylogenetic similarities. It's best that we look at each case on its own merits and try to figure out what's going on, rather than trying to lump them all together under the rubric of intelligence. In hominids, proboscids, and cetaceans, where we had the best data, there was a long period of stasis followed by rapid change. And this is what I want to talk about for the rest of the talk, is this period of stasis followed by change. I think I've shown you that in all the 3 lineages, that there was a stasis and then rapid change. But we have to go back to some of the physiology of the brain. And this is something that's actually often ignored by people. Is that mammalian brains produce their own heat independent of the body. We don't keep our brains warm because we're generating heat in our bodies. Now, obviously, the brain can't produce heat through shivering thermogenesis, but they do produce heat. And this is a very rough sketch of an experiment that was done by a Hungarian guy in the 1970s, where he took a rabbit and measured its brain temperature and body temperature for a while, then buried the rabbit up to its neck in ice and measured brain temperature and body temperature. Brain temperature stayed the same, body temperature started to drop off quite dramatically. So this indicates, and has since been proven, that the brain produces its own heat. And this is a very, very important aspect biologically. So what produces heat? In the body, we think about non-sugary thermogenesis, and we think brown fat. Okay? There's special proteins in the cells of brown fat that are found inside the mitochondria. And these are called uncoupling proteins. So they're in a mitochondrial membrane. They dissipate the protein gradient in the mitochondria before it can be used to provide energy for oxidative phosphorylation. So what they do is they uncouple the ATP cycle, and all the energy that was going to go into the ATP cycle to run the normal functions of the cells gets released as heat. So they were discovered in brown fat, and there's 5 different types of uncoupling proteins. So there's been a few papers that have shown that uncoupling proteins are found in the brains of rats, and people have kind of left it at that. But to me, this is a really, really interesting idea. If I was to be right about the cetaceans evolving a large brain for thermogenesis, they must have specializations to produce more heat in their brain, because they're under pressure of losing heat. So I followed this up, and over a glass of Amarula in Pilansberg Game Reserve, I was talking with a friend of mine, Shel Fuchser. He was the guy who discovered the catecholamine systems in the brain before I was born. And he told me about these uncoupling proteins, and we should look for them in the brains of these different animals. So we did. And this turned out to be very nice. We're looking at, so there's 5 types of uncoupling proteins, 1, 2, 3, 4, 5. They're kind of easy to remember in that sense. We first started looking at artiodactyl cerebral cortex, because cetaceans are part of the same mammalian order as artiodactyls. You see here in this little image of the lower half of the cerebral cortex. So here's white matter, this is layer 6, layer 5, layer 4, and so on. And this is in the sand gazelle, and this is in the dromedary camel. And we see the location of neurons marked immunohistochemically that are full of uncoupling protein 1. We look at a few more, there's 2 images here, it gets me confused. We look at a few more, this is the springbok, this is the kudu, again towards the bottom of the cerebral cortex, and we see that there's neurons that have this uncoupling protein in them. Not all of the neurons, it's about 30% roughly that have uncoupling protein 1 inside of them. So that's kind of interesting. These neurons are doing neuronal things, but they also have this possibility that they can be co-opted to produce heat instead of doing things that neurons are supposed to do. So that was nice. That sort of set the baseline. We looked at about 12 different artiodactyl species of various sizes, and across them it was always kind of the same thing. So then we looked at a harbor porpoise. And this is slightly higher magnification. You see the neurons here and lots of glia in between. We see the neurons staying with uncoupling protein 1 here in the harbor porpoise. And it's about 70% of the neurons have uncoupling protein in them. So twice as many as what we see in artiodactyls. We looked at the minke whales. I was lucky enough to go on a whaling ship and get brains from a minke whale. So that's another story altogether. 100% of neurons in the brain of a minke whale are immunopositive for uncoupling protein 1. If we look at a humpback whale, it's some tissue that was sent to me by a friend who got the humpback whale brain. Again, 100% of neurons have uncoupling protein 1 in them. If we look at a summary of this across the species we've looked at, we see here are all the artiodactyls ranging from the small sand gazelle brain, domestic pig, the Nubian ibex, springbok, blessbok, kudu, I think blue wildebeest, camel, nyala, the hippo, which is the closest living relative, cetaceans, and the buffalo. All of them have around 30%. And this is the average here with the the standard error for artiodactyls. And this is in primary visual cortex and anterior cerebral cortex. So we looked at two different parts of the brain, one that's involved purely in sensory processing, one that's involved in the higher, what's thought to be involved in higher cognitive processes. And over here, we see the cetaceans. So we see that the average difference is very high. So the cetaceans have a way of producing heat that's special compared to other mammals. It's not different, there's just more of it. So there's a real quantitative change in the ability of cortical neurons to produce heat in cetaceans compared to their artiodactyl relatives. But I wasn't happy with just that. There had to be more there. So we started doing staining for all the other different uncoupling proteins, and some of them didn't work, but some of them worked really, really nicely. Here we have the cerebral cortex from a Harvard porpoise stained in missile. And here we have glial cells immunoreactive for uncoupling protein 5. Here in the white matter, it's just glia, there's no neurons there. And here we have glia immunoreactive for uncoupling protein 5. So about 40% in the cortex, about 73% of glia in the white matter. So there's a lot of unusual staining of uncoupling protein in glia in the cetaceans. If we look at the other species, the minke whale, about 40% in cortex, 55% in the white matter, so a little less than the Harvard porpoise. And if we look at the humpback whale, again, we see about 30% of glia in the cortex, 60% of glia in the white matter are immunoreactive for uncoupling protein 4 and 5. So I've used 4 and 5 interchangeably here. The results are the same between the two, but these are just the nicest pictures to show. Excuse me. However, uncoupling proteins are found in all mammalian brains, and they're found for heat production. So this is the Western blot, the Harvard porpoise, minke whale, the humpback whale. Here's a range of different artiodactyls, and so you can see the uncoupling protein 4 is there. There's the cetaceans again. More of the artiodactyls. On the end here is the laboratory rat. So you see that all these animals have uncoupling protein 4 in the brain, but it's only in the cetaceans that it's found within the glia, both within the white matter and within the grey matter. So the other thing that's really interesting is, how is this uncoupling protein activated? And this is something we only found the other week. And I was talking with some guys from Germany that heard me give a talk a couple of years ago, and they emailed me and they sent me, they worked on brown fat, so they sent me one of their reviews, and in it was something that really struck a chord with me. It appears that in brown fat, noradrenaline is the molecule that stimulates the activity of uncoupling proteins and short circuits the ATP cycle. So when we look at cetaceans, when we stain the noradrenergic neurons, I've done this with tyrosine hydroxylase, we're going to deal with more specific staining soon, we see the number of boutons and axons within the cerebral cortex of the minke whale is incredibly high, and within the white matter. Now in other animals you just don't find this. The axons for TH are usually only located in the upper superficial layers of the cortex, and you definitely do not find any boutons in the white matter of other animals. So this is really unusual. So they have a specialized noradrenergic input to stimulate the uncoupling protein in the glia to produce heat. So cetaceans do have two really specific specializations for increasing their ability to produce heat within the brain. And they use this ability to overcome heat loss to the water because they're living in very cold environments. Okay? All these adaptations probably evolved at the same time when the brains got large, because there was a massive drop in water temperature. So large brain cetaceans evolved as a response to thermal selection pressure. So I think that I'm pretty comfortable with that explanation. I have some friends doing some genetic work on this to look at the expression of the uncoupling protein genes and doing real-time quantitative PCR and then a whole lot of fancy molecular stuff that I have no idea about. But the results that are coming in so far support what I've found with my more simple techniques and approaches. Now, but what about other large brains? We thought, okay, there's elephants and there's humans, and I have tissue of this in the freezer. Let's look at the uncoupling proteins in these guys. So we took out some blocks, we started staining them. Uncoupling protein 1 in humans and elephants, 40 to 45% of the neurons were immunopositive for uncoupling protein 1. 2, 4, and 5, we got no findings for. Nothing was staining the brains of humans and elephants. And we looked at chimpanzees and baboons and macaque monkeys and hyraxes and manatees, all the relatives of the elephants and the humans. We didn't really find much at all until we looked at the uncoupling protein 3. This is the white matter of the elephant, so this is several millimeters below the surface of the brain, and about 60% of the glia are immunoreactive for uncoupling protein 3. None of the glia in the cerebral cortex of the elephant is immunoreactive for uncoupling protein 3. It's just in the white matter. And we looked at the human, and about 9% of the glia has uncoupling protein 3 in it as well. So this is really unusual. All these three brains, or the model representatives of these groups that had a long period of stasis before evolving large brains over 700 grams, all have uncoupling proteins in them, and specialized in the glia as well. And that's really, really interesting because the mammalian brain produces its own heat, but that's based on the rules of how mammalian brains produce heat when mammals evolved a long time ago with really small brains. And it seems that that works nicely up to about 700 grams, and then you need something a little bit extra to get a larger brain than that. So I kind of picture in my head that in the hominids, they had this sort of scaling that if their bodies got bigger, their brains could get bigger. But it just wasn't going to work. It just wasn't going to work until the advent of uncoupling protein 3 within the white matter. Now, what will that do is the larger brains, brought about through independent evolutionary trajectories, have larger white matter tracks. And so perhaps what's needed is additional heat in these white matter tracks to maintain the ability of neurons and axons to project over a long distance and maintain their patency and keep sending action potentials backwards and forwards. So when a brain is larger than the standard thermogenetic capabilities of a mammalian brain, you need to add something to it. Okay? So why do large brains evolve? The uncoupling proteins appear to be a very distinct metabolic key that unlocks and permits the evolution of large brains. The large brains don't evolve for intelligence, but for other reasons. The majority of brain size evolution can be explained by imperatives of allometric scaling, and we see that in human, elephant, pinnipeds, camels, although they're different trajectories, or in response to allometry and a specific selection pressure, which is the thermal selection pressure in cetaceans. So there's no reason to think that large brains evolved for intellectual capacity. I think that just came along afterwards in some lineages, but not in all lineages. And that we can actually go to a much more simple explanation in that increases in body size with one metabolic key allowing for large brains to evolve being the real answer as to why large brains came around. So most of this work has been funded by the South African National Research Foundation, so I should give them credit. And I want to thank all the students and collaborators, past and present. They all know how much work they've done and how much help they've been to me and how much they mean to me. Thank you very much.